Hello friends and welcome. Today we will be talking about the physicochemical properties of drugs. The main objectives of today's deliberation will be to introduce various physicochemical properties of drugs needed for drug formulation, to know the concept of drug dissolution and its importance in drug designing, to get familiarized with the importance of partition coefficient of drugs, to understand the concept of bioavailability and its importance to drug chemists, and finally to understand the acid-base properties of drugs. First of all, let's know what drugs are. What do we mean by physicochemical properties of drugs and what's their importance in the drug formulation and designing? Well, drugs belong to the category of the chemical substances that are employed either to prevent or to cure diseases in humans, animals, and plants. They are rarely administered solely as pure chemical substances, but instead are almost always given in formulated preparations. Drugs can vary from relatively simple solutions to complex drug delivery systems through the use of appropriate additives or excipients in the formulation to provide varied and specialized pharmaceutical functions. The main objective of dosage form design is to achieve a predictable therapeutic response to drug included in a formulation which is capable of large-scale manufacture with reproducible product quality. Therefore, designing of a drug not only involves chemists but also workers from a wide range of disciplines such as biology, biochemistry, pharmacology, mathematics, computing, and medicine, amongst others. Besides, to address the problem of high iteration rates and in keeping with good management practice, analysis of the entire drug discovery process has been essential to help drug companies compete in the current environment. Medicinal chemists have also been actually involved in understanding drug failures by examining and defining the physicochemical properties of compounds that predict successful outcomes. The aim of such analysis is to improve the quality of compounds that enter clinical trials, and yet there is some debate to the extent these guidelines are being followed. Appropriate and efficacious dosage forms will be prepared only when all these factors are considered and related to each other. So, drug formulators need to take many considerations while designing a drug. Among many other factors, dissolution rates, partition coefficient, acid-base properties, and bioavailability are important parameters a formulator must assess while designing a drug. So, in this topic, we will be considering each one of these properties separately. First, let's come to drug dissolution. For a drug to be absorbed, it must be first dissolved in a fluid at a site of absorption. For example, an orally administered drug in tablet form is not absorbed until drug particles are dissolved or solubilized by the fluids at some point along the gastrointestinal tract, depending on the pH solubility profile of the drug substance. So, drug dissolution is the process by which drug particles dissolve in the body fluids. Hence, drug dissolution rate is defined as the amount of drug substance that goes in the solution per unit time under standard conditions of liquid-solid interface, temperature, and solvent composition. Now, let's understand the mechanism of drug dissolution. The mechanism of drug dissolution is quite simple. Drug molecules in the surface layer are first broken into small granules which are further disintegrated into even smaller particles just to increase the surface area for dissolution. These smaller particles then dissolve in a way that the solvent forms a saturated solution around the particles to form a diffusion layer. Dissolved drug molecules then keep on passing through the dissolving fluid to come in contact with the mucosa, where they are finally absorbed. Replenishment of diffusing drug molecules in the diffusion layer is achieved by further drug dissolution, and the absorption process continues. Mechanism of drug dissolution is explained by diffusion layer model, or also known as film theory, which proposes that dissolution of a crystal immersed in a liquid takes place without involving reactive or electrical forces. It explains drug dissolution in two consecutive steps, with solution of solid forms a thin film or a layer at the solid-liquid interface called as stagnant film or diffusion layer saturated with drug. This step is usually rapid, or more precisely, we can say it's instantaneous. The second step involves diffusion of the soluble solute from the stagnant layer to the bulk of the solution towards the mucus. This step is slower and hence is the rate-limiting step in the drug dissolution. If the dissolution is fast or the drug is delivered and remains in the solution form, the rate of absorption is primarily dependent upon its ability to traverse the absorbing membrane. If, however, drug dissolution is slow due to its physicochemical properties or for other formulation factors, then dissolution may be rate-limiting step in absorption and can influence drug bioavailability. The dissolution of a drug is described by general Noyes-Whitney equation. dm by dt is equivalent to Ka Cs minus C, where dm by dt is the dissolution rate, K is the dissolution rate constant, A is the surface area of the dissolving drug, Cs is the concentration of the drug in saturated diffusing layer, and C is the concentration of the drug in dissolution medium at time t. This equation reveals the dissolution rate can be raised by increasing the surface area, that is, by reducing particle size of the drug, or by increasing the solubility of the drug in diffusing layer, and by increasing K, which incorporates the drug diffusion coefficient and diffusion layer thickness. During the early phases of dissolution, Cs is greater or equal to C, and if the surface area A and experimental conditions are kept constant, then K can be determined for compacts containing drug alone. The constant K is now termed as intrinsic dissolution rate constant and is characteristic of solid drug compound in a given solvent under fixed hydrodynamic conditions. For the drug designers and formulators, the property of drug dissolution is important to determine before formulating a particular drug. This is because dissolution rate, when combined with other drug factors, provides an insight to the formulator into the potential in vivo characteristics of a drug in question. Drugs with values of K below than 0.1 per mg per centimeter square usually exhibit dissolution rate limiting absorption. Particulate drugs can also be examined where an effort is made to control A and formulation can be studied. However, in vitro tests only have significance if they can be related with in vivo results. Once such a relationship is established, in vitro dissolution tests can be used as quality control tests. Nevertheless, the importance of dissolution testing has been recognized recently by official compendia with the inclusion in recent editions of dissolution specification using standardized testing procedures for a range of official preparations. After drug dissolution, now let's come to another important property of drugs, that is partition coefficient of a drug molecule. Partition coefficient in pharmaceutical sciences is used to estimate the partition of drugs within the body depending upon the degree of hydrophilicity of drug in different phases. As we know, a cell membrane has a central layer that's lipophilic, or we can say hydrophobic. By lipophilic, we mean having an affinity for fat and high lipid solubility, a physicochemical property which describes a partitioning equilibrium of solute molecules between water and an immiscible organic solvent favoring the latter. The relative solubility of a substance in water and an organic solvent, water immiscible, is a measure of lipophilicity. Experiments have shown that lipophilicity is an important property of a drug that influences passive transport across a cell membrane. A drug molecule needs to be sufficiently lipophilic so as to enter a lipid core of a cell membrane, but not so lipophilic that it's permanently retained in that core and does not reach its actual target. In drug discovery and development, lipophilicity is usually expressed by the partition between water and octane 1-ol. The partition coefficient P of a drug is given by P is equivalent to concentration of a particular drug in octanol divided by concentration of the same drug in water. Drug formulators determine partition coefficient mostly by shake flask method. The method, which involves shaking the drug sample with a mixture of octane 1-ol and water and then determining its concentration in each layer spectrophotometrically. Hydrophobic drugs with high octanol to water partition coefficients are mainly distributed to hydrophobic areas such as lipid bilayers of cells. Conversely, hydrophilic drugs with low octanol to water partition coefficients are found primarily in aqueous regions such as blood serum. Sometimes the value of partition coefficient is calculated from the knowledge of molecular structure rather than experimentally. It must be noted that partition coefficient is different from distribution coefficient. Partition coefficient generally refers to the concentration ratio of an ionized species of compound, whereas distribution coefficient refers to the concentration ratio of both ionized plus unionized species of the compound. Coming to the application of partition coefficient in drug chemistry. A drug's distribution strongly affects how easily the drug can reach its intended target in the body, how strong an effect it will have once it reaches its target, and how long it will remain in the body in an active form. Hence, partition coefficient of a molecule is one criterion used in decision making by drug formulators in preclinical drug discovery. As already discussed, for a drug to be orally absorbed, it normally must first pass through lipid bilayers in intestinal epithelium. For efficient transport, the drug must be hydrophobic enough to partition into lipid bilayer, but not so hydrophobic that it will be retained by the lipid bilayer. So, partition coefficient of a drug molecule plays a major role in determining where drugs are distributed within the body after absorption, and as a consequence determine how rapidly they are metabolized and excreted. Now coming to the third important property of drugs, that is bioavailability. One of the principal pharmacokinetic properties of drugs is bioavailability, which can be defined as a subcategory of absorption and is the fraction of an administered dose of unchanged drug that reaches the systemic circulation. By systemic circulation, we actually refer to venous blood or arterial blood, which carries the intact drug into tissues. Certain dose of a drug administered to a patient will give no guarantee that all of the dose will reach the systemic circulation, but with the exception when the drug is administered intravenously. The fraction of the drug that actually reaches the systemic circulation, and that too in unchanged form, is known as bioavailable dose. Bioavailability is thus concerned with the quantity and rate at which the intact form of drug appears in the systemic circulation following oral administration of that drug. Bioavailability exhibited by a drug is hence considered very important in determining whether a therapeutically effective concentration is achieved or not at the site or sites of action of the drug. Coming to the types of bioavailability. Bioavailability is of two types, namely absolute bioavailability and relative bioavailability. First, coming to absolute bioavailability. The absolute bioavailability of a given drug from a dosage form is the fraction or percentage of the administered dose which is absorbed intact into the systemic circulation. Absolute bioavailability may be measured by comparing the total amount of intact drug that reaches the systemic circulation after administration of a known dose via an absorption site with the total amount of the same drug, same dose that reached the systemic circulation after intravenous administration. An intravenous bolus injection of the drug is used as a reference to compare the systemic availability of the drug administered via an absorption site, since in the former case the entire administered dose of the drug is introduced into the systemic solution and is considered to be totally bioavailable. The absolute bioavailability of a given drug using blood plasma data may be calculated by comparing the total areas under plasma concentration time curves obtained following the administration of equivalent doses of same drug via an adsorption site and via the intravenous route to the same subject on different occasions. So, in simple terms, for equivalent dose of drug administered, absolute bioavailability is equivalent to total area under plasma concentration time curve following drug dose given at absorption site to the total area under plasma concentration time curve following drug administration by rapid intravenous injection. Now coming to another type of bioavailability, that's relative bioavailability. In case of drugs which cannot be administered through intravenous bolus injection, the relative or comparative bioavailability is determined. In this case, the bioavailability of given drug from a test dose form is compared to bioavailability of the same drug administered in a standard dosage form, which is either an orally administered solution of the drug or an established commercial preparation of proven clinical effectiveness. So, bioavailability measurements are often used to determine the effects of dosage form. It measures the effectiveness of a given drug It's actually the bioavailability which determines the actual amount of absorbed dose and hence is the most important criterion determining the success of a drug. Having understood the concept of bioavailability, now let's switch on to another important property of drugs, which is acid-base property. The acid-base properties of a molecule are among the most fundamental for drug action. However, they are often overlooked in a prospective design manner unless it has been established that a certain ionization state, for example, quaternary base or presence of a carboxylic acid, appears to be required for activity. It's fairly common for drugs to be classified as weak acids or bases. Upon dissolution in water, drug molecules ionize to different degrees depending on their chemical structure. Unlike strong acids like nitric acid or strong bases like sodium hydroxide, which ionize completely upon dissolution in water, weak acids or bases will exist in an equilibrium of ionized or unionized species. The ionized form will include either cations or anions. Therefore, the ionization of many drugs in the aqueous solution of body fluids is an equilibrium process. Drug molecules are weak acids or bases in physiological fluids by virtue of functional groups in their chemical structure. That is, acidity or basicity of a drug molecule is dependent on having electron-poor hydrogens, for example, carboxyl group, or non-bonding electrons which can be donated to hydrogen, for example, those on amine nitrogens. Some of the functional groups most commonly found in drug molecules are keto, aldehyde, amide, azo, carboxylic, ether, ester, phenolic, hydroxyl, etc. According to Bronsted and Lowry acid-base concept, an acid is a substance that can donate proton, while a base is a substance that can accept proton. Many drugs are formulated as salts, a process that involves taking the weak acid or base and reacting it with base or acid respectively in order to generate an ionic compound or salt. When salts are dissolved in water, the ions dissociate completely and associate with water molecules to form solvated anions and cations. One common error is to confuse low solubility with low % dissociation, but these two processes are totally different. Barium sulfate, for example, has a very low water solubility, but whatever amount does dissolve is 100% ionized into barium 2 positive and sulfate 2 negative ions. The same applies to many drugs formulated as salts. They may have varying degree of water solubility, but whatever amount is dissolved in water is 100% ionized into the component ions. Indeed, the decision to formulate a drug as a salt, usually adding one more step in the manufacturing process, comes from the need to obtain greater solubility in the body fluids. This salt is invariably more soluble than the parent compound, although with some exceptions. So, acid-base properties are important in determining the solubility of the drug in body fluids and determining the rate of action of the drugs. To conclude, the main objective of dosage form design is to achieve a predictable therapeutic response to drug included in a formulation which is capable of large-scale manufacture with reproducible product quality. For a drug to be successful, a formulator must first consider all the properties of the drug related to physical and chemical structure. These physicochemical properties include drug dissolution, partition coefficient of drugs, bioavailability, and acid-base properties of the drugs. Knowing these properties in advance to drug formulation can help the pharmacologists to design successful drugs without investing a lot of synthesis, time, and money. This is all about today's lecture. Hope you have understood it well. Thank you and goodbye.